The Microcosm of Mission Bay, San Diego by Michael Fig. All right, so there has been a sewage crisis along the U.S. Mexico border. This involves the country of Mexico's Tijuana River, which has carried millions of gallons of untreated sewage, industrial waste, and urban runoff into San Diego County's coastal waters. This has led to health concerns and environmental degradation and public outcry in addition as well in protest against this. Public outcry has occurred actually more in the United States than in Mexico. This is due to infrastructure failures of Mexico's wastewater treatment facilities that cannot handle Tijuana's rapid population growth. And this is caused sewage spills. So this also led to health risks, water contamination, including harmful bacteria, viruses, and antibiotic-resistant pathogens. Also resulted in beach closures, and some beaches in San Diego County have been closed for up to 1,000 days due to the pollution. There has been, thankfully, some cross-border cooperation between the United States and Mexico, and they're working on a permanent solution via the environmental protection agencies and their projects. Next slide. All right, so if there's any time considerations that you're not able to view this mini documentary on the very same subject I just mentioned, you can move on to the next slide. All right, with this with this microcosm sample of Mission Bay, we can learn about how sensitive an ecosystem can be in response to unnatural interference. Uh, So a sealed microcosm can be self-sufficient with its own resources. The sample taking, which is shown right in this area, encompasses approximately 25 feet by a 25-foot area. All right, my hypothesis is as follows. The microbial community and plant health in the Mission Bay area are significantly altered by the presence of contaminants from Tijuana, Mexico, sewage runoff, leading to changes in soil and nutrient availability, water quality, and plant growth. Next slide. All right, so the sample site I chose is between Playa Pacifica Park and Mission Bay Resort. So during a walk with along the area, I chose a site that had calm waters and a decent material ecology, which would be defined as the interplay between natural and natural elements, also reflected in this ecosystem here. Um, some materials that I used in this specific spot are about 175 grams of sandy loam soil. It did have a good portion of clay. Also had 200 milliliters of bay water, and there was some sea algae that washed ashore that was visible upon the soil. There's a nearby monocot grassy plant that I included, and when I brought it home, quickly added 15 to 20 chia seeds to test to see if there any germination could uh, occur and be viable in this environment. The duration is for 46 days of this project, and the temperature ranges from approximately 15 degrees Celsius to 25 degrees Celsius each day. The container I used was an airtight mason jar with a clamp seal. Capacity is about 1,000 milliliters or about 34 fluid ounces. Next slide. All right, so some observations and data from week one. The sediment strata settled in under 24 hours. Light conditions included part sun in the morning and indirect light in the afternoon. And initial color by strata is dark brown sandy loam soil. Clear water, green algae, and sea moss were still visible, and then one monocot grassy plant was included. No other forms of visible life were swimming around. Bacteria, fungi are naturally assumed to be present in both the water and in the soil. And there's, of course, like I mentioned before, is a quick inclusion of 15 and 20 chia seeds used to test the habitat viability. Changes in week two, the water had become visibly cloudy, like gray in color. Algae and sea moss are difficult to preserve. Difficult to preserve, but the green model plant was also difficult to preserve already. The chia seeds had not germinated. Some week three changes, the water become uh, slightly darker, but still light gray in color. Slightest hint of pink hue in the water. Algae and sea moss were difficult to observe, as well as the monocot grassy plant, and the chia seeds still had not germinated. By week four, the changes became slightly more dramatic, with a light pink color with very small hints of green at the surface. The algae and sea moss were very difficult to observe at this point, as well as the monocot grassy plant. And at this point, all plant life went brown in color, and we could safely say at this point that chia seeds were past the terminal germination date. In week five, the pink color in the soil became a more reddish than pink color with a hint of light green growth at the surface. Um, so the light green was a little more visible. Algae and sea moss were, of course, difficult to observe, as well as the monocot grassy plant. All plant life still appears brown. Chia seeds are still past their terminal germination date. In week six, more changes have occurred. Water became a deeper red color with a majority of green growth at the surface. My approximate uh, observation was that the green growth at the surface was about 80% at this week and 20% red, and algae and green moss, of course, were difficult to observe. They must have transformed, and including the monoclot grassy plant and chia seeds are still past the terminal germination date, so I would not expect anything else to happen with them at that point. All right, and then changes to week seven. The water became a deeper red wine color. The majority of the green growth at the surface, and it was difficult to see the color of the water through the surface. Approximate percentage of the green growth at the surface was 95%, and red water was only visible about 5%. And the algae and sea moss, of course, were difficult to observe at all. Green mossy, green the grassy monocot plant would turn brown, and it was so difficult to observe. And the chia seeds were still, of course, past their terminal germination date. All right, and I also created a GIF animation showing the progression of the composition and color and texture throughout the 7 weeks, and a color chart showing the color change progression by strata from week 1 through week 7. Soil being the lowest strata and the water surface major, or minor being the surface of the water. Minor meaning the water was water color at the surface whatever majority color I observed, and major being the majority color observed. And of course, the plants went from green to brown progressively. All right, so my major findings in regards to my hypothesis, although the nature of the color change was not expected, the change in the water appearance corresponds with Southern California's ocean contamination. I did not expect the red color to occur, but I did expect something to happen in terms of color change or water change. My hypothesis predicted poor water quality and plant growth, which is what we got, and such was revealed over the time. The appearance was pink and red water. Appearance of pink and red water compelled me to investigate further into known causes. One of these uh, conditions that led to this change is apparently called eur- euro- eutrophication, where you have excess of nutrients in the water, increasing the ability of the cyanobacteria to fix nitrogen. I observed my submerged plants did not survive, and the biodiversity of microcosm was dominated by microorganisms. These microorganisms could be connected to waterborne diseases. This finding did not lead me to an answer to the color changes, so I investigated further. Next slide. The likely cause, according to the National, National Oceanographic Association fisheries, is the upwelling of deep ocean water, leading to the toxic or harmful green algae blooms, also known as HABs. The ability to observe the phenomena clearly occurred in week 5 of my microcosm. Seems to correspond quite well, and is further supported by the observations by the NOAA fisheries, who observed and helped marine mammals that became sick by a neurotoxin associated with HEBs known as domoic acid. Beachgoers are also warned to stay clear of these mammals. NOAA is also researching and taking care samples to investigate. I had not yet found a reason for the red color of the water in my microcosm, so I investigated further. According to the Science Direct website, the conditions that were necessary for the bacteria known as Serratia marcescens may have been present in my microcosm due to the red pigment in that strain and their ability to break down DNA. In my case, plants turned brown and biodegraded. Next slide. Other research findings have noted that this particular strain of Serratia exists in an animal feces, dust, soil, and surface waters. Additionally, the bacteria grow in areas such as soap residues in bathing areas, feces in toilets, and soap and food residues. The secondary source of and location appear to correspond to the sewage runoff of Tiajuana River and San Diego Ocean and Bay. Yeah, so you can see that red color in this bacteria magnified. That uh, corresponds to the conditions and the color. Um, in conclusion, I have 5 main points I wanted to state. One, my microcosm demonstrated apparent eutrophication that accelerated the algae growth at the surface of my microcosm. Secondly, microbial succession apparently caused a chain reaction of bacterial growth in my sealed microcosm, leading to appearance of Serratia marcescens and a toxic green algae at the surface. The sewage waste runoff of Tiajuana River also likely contributed to the microcosm color changes and to the harmful aquatic and marine mammal ecosystems. All right, and fourthly, the neurotoxin known as domoic acid in the Southern California waters affected marine mammals in the higher trophic levels. And lastly, being self-contained and self-regulating, the ability of the microcosm nature to balance and correct the excess bacteria that leads to eutrophication, and the growth bacteria of bacteria such as Serratia marcescens reveals a concentrated sample of the current ecological dynamics of San Diego waters. For further research, I probably would attempt another microcosm samples at areas that have more moving water, such as the beaches at Mission Beach and Imperial Beach. If the plants were available and I did not have any time constraints, I'd also add more plants if they're available that are more able to access the microcosm CO2 air pocket. And this comparison would enable an hypothesis that could demonstrate the benefit of more water flowing through the ecosystem at a beach versus the Bay Area. All right. And thank you for listening. And if you have any questions or comments, concerns, please say them for the feedback area.